The method that was used was vitro studies with human adipocyte and pre-adipocyte. The sample was obtained by isolation of adipose tissue from healthy female between the ages of 25 to 60 years. The adipose tissue was washed with hand gland sauce, and it was centrifuged. Then centrifugation for 5 minutes was done at 380 J. The floating tissue was decanted into 1 ml medium A, which we will look at what it contains later on. This was constantly shaken and filtered with nylon filter. After centrifugation for 10 minutes, it resulted into separation, which was stroma, vascular palate, and floating adipocyte. The adipocyte was suspended in medium B and C. 5 minutes centrifugation was done at 200 J for 5 minutes, and later on it was snapped in a frozen ice. Then the stromovascular palate was suspended in 100 ml of red blood cells, lysis buffer for 10 minutes. Centrifugation was done for 10 minutes at 380 J. Then the palate was suspended in medium D, filtered with nylon mesh. The next adherent pre-adipocyte was washed 3 times with medium E and cultured for 24 hours. Pre-adipocyte was suspended to differentiate flags for 16 days in medium F. Medium weight changed every 3 days. Pre-adipocyte medium was then fractionized, then washed with phosphate saline. The palate, it was then palated for 10 minutes at day 0, 1, 2, 4, 8, and 16. This was stained by oil red at the first day. And this gives what we had in the following medium. As medium A, we can see medium B is different from the difference between medium B and A is that A is that collagen, and we can see medium C contains
of the neurin resistin gene was expressed in both human adipocytes and pretocytes. However, although resistin transcripts were barely detectable in primary cultured mature adipocytes, expression levels were 55-fold higher in primary culture undifferentiated pre-adipocytes. As you can see in this graph here, differentiate expression of the resistin gene in human adipose cells after isolation of pretocytes and adipocytes from human adipose tissue. Cells were cultured for 24 hours in identical serum-free medium. And obviously this is the relative expression of the resistin gene just across here, and this is the adipose tissue and the pretocyte tissues. From the results, as it was expected from the findings, the adipogenic differentiation was associated with time-dependent reduction in resistin gene expression. As we see here, as you go across the days, the expression does reduce just here. As we go up here, this is the gene expression, and this is the days. On day 16, you can see there's a lot less, whereas on day 16, there's a lot more. This just shows the relationship between resistin gene expression and human adipocytes in insulin resistance. This is the actual gene resistant, and this is the expression and insulin sensitivity. Therefore, we can say that